Science is the chief defense of the nations and is the transmission of civilization. We have here done our bit to strengthen our own belief in science and its contribution for animal and human welfare. Welcome to Ome Research Facility, a research facility for chromosome, proteome, transcriptome, and genome analysis, each sounding and vibrating like Om, which is something very. Department of Animal Biotechnology is an offshoot of Department of Animal Genetics and Breeding. It was initially established as a part of AGB in 1999 and emerged as a separate department in the year 2000. Vision of this department is understanding life by exploring its intricacies, and mission is to improve production, reproduction, and health of animals through biotechnology. The thrust areas of research are genomics, disease diagnosis, vaccine production, artificial insemination, embryo transfer technology, pharmacogenomics, nutrigenomics, surgery, and let us have few glimpses of the research endeavor and achievements so far. Whole genome sequencer FLX is a pride possession of this department. It's a workhorse with a capacity to produce 1 GB data in a day. Whole genome sequencing of Jafrabadi buffalo was carried out by this department for the first time in the world. The resulting 64,212 contigs were deposited at GeneBank. We have also taken up various projects in collaboration with other institutions and organizations for whole genome sequencing of various pathogenic microorganisms like Pasteurella maltosida, plant viruses, and non-pathogenic microorganisms like Acidiopsillus ferrooxidans, Bacillus amyloliquefaciens, Rhodococcus species, Panibacillus species, Lactobacillus rhamnosus, and Lactobacillus helveticus. We have also carried out transcriptome analysis of horn and buccal cancer, biotic stress in plants, as well as metagenomic analysis of rumen microbes in buffalo, goat, and camel, poultry gut microbes, as well as microbiota of subclinical mastitis in gyr, kankaraj, and different markers related to production, reproduction, disease resistance, and growth were also studied in various breeds of livestock and poultry. To estimate the genetic diversity within and between the breeds, as well as to understand the genetic structure of the different livestock breeds, we have selected microsatellite as a choice of genetic marker. We have carried out microsatellite based genetic characterization of gyr, kankaraj, kankatha, and nimari cattle, Jafrabadi, Mehsani, and Surti buffaloes, Surti, Jhalawadi, and Gohilwadi goats, as well as Kathiawadi horse and an achievement that has direct field application is meat species identification by PCR based technology, which also involved differentiation of cattle and buffalo meat. Patent application has been filed. We have studied gene expression profile to understand the lactogenesis in various models of buffaloes, mammary tumors, and high FCR bird. We have also standardized the molecular methods for quantitative and qualitative disease diagnosis for various genetic, bacterial, and viral diseases like ehrlichiosis in lions, tuberculosis, hemorrhagic septicemia, brucellosis, and bovine herpes virus 1 in bovines, Marek's disease, infectious bursal disease, and chicken infectious anemia in poultry, canine parvovirus and progressive retinal atrophy in dogs, and pest pedipatids. We are extending our support to human diagnostics also, particularly human leukocyte typing of donor and recipients for kidney transplantation. We are also providing services for parentage verification in Mehsani buffalo under field progeny testing program for accuracy of data for Dudhsagar dairy. In nutrigenomics, for the first time in India, we have characterized rumen microbes using 16S rRNA and protozoa using 18S rRNA. Surgeons have come out of operation theaters seeking help of biotechnology for some of their surgical affections. Progressive retinal atrophy is one such condition where dogs become blind. Similar conditions also exist in human being. We have confirmed mutations causing PRA in Spitz and Cocker Spaniel. Bioinformatics is a tool very much essential in the field of biotechnology. We have developed two softwares. One is GoTLay, used for breed identification among Jhalawadi, Gohilwadi and Surti goats, whereas Kamdhenu software for breed identification among Gir, Kankatha, Kankrej, Malvi, Nimari and Galau cattle. This identification is based on several research projects have been funded by number of funding agencies like Government of Gujarat, Gujarat State Biotechnology Mission, ICAR, NATP, NAIP, DBT, DST, Murjibai Patel Urological Hospital Nadiyad, Maharashtra Livestock Development Board and Rashtriya Ome Research Facility, where scientific pursuit is attaining new heights, where modern education is being defined and practiced, and of course, BT, biotechnology, is being witnessed, providing the momentum for Bharat